So we are now moving to the talk of Li Chan from London, and she will talk about ways of androgen decrease, and the theme is around ACTH inhibitors. So please, the floor is yours.
Thank you so much for the organising committee to invite me to speak today. So the title of my talk is Ways of Androgen Decrease, with a focus on ACTH inhibitors. And I think that it comes very nicely after Heidi's talk about TARTs. So I am Li Chan. I am a consultant paediatric endocrinologist based in London. So this is a diagram of the hypothalamic pituitary adrenal axis. So this is the axis that controls the production of glucocorticoids. So this is cortisol in humans, as well as it controls the adrenal androgen production. So CRH, corticotrophin releasing hormone, and ABP, arginine beta-pressin, are hormones that are secreted in the hypothalamus that acts on the pituitary gland to produce ACTH. This hormone then gets secreted in the bloodstream and acts on the adrenal gland to produce cortisol. Cortisol then negatively feeds back on the pituitary and hypothalamus to stop further production of ACTH. So in the case of congenital adrenal hyperplasia, where there is an enzyme problem, this means that little or no cortisol is produced from the adrenal gland. The result of this is that there is no negative feedback. The pituitary senses a lack of cortisol, and this will then produce an excessive amount of ACTH to try and increase the cortisol production from the adrenal gland, which is not possible due to the enzyme block. However, this excessive amount of ACTH results in the production of adrenal androgens, such as androstenedione, DHEAS, and testosterone. So as you have heard previously, too much androgens can have lots of problems associated with this. So this ACTH excess is a real problem, which includes virilization, hirsutism, short stature and adulthood, precocious puberty, and the development of TARTs. So currently, the physiological treatment of CAH is hydrocortisone or other forms of steroids in a replacement dosage scenario. But this is often inadequate to suppress the ACTH excess, leading to, if people try and increase this dose, this leads to excessive glucocorticoid exposure in the long term. So this is a real problem and an unmet clinical need. So excessive androgens and excessive amounts of glucocorticoids being used to suppress this ACTH contribute to the long-term health problems patients face with congenital adrenal hyperplasia. Here are some of the publications highlighting how patients with CAH may have adverse health outcomes, such as increased likelihood of obesity, high lipid levels, high blood pressure, impaired fertility and TART formation, as the previous speaker has mentioned, adverse quality of life, as well as effects on brain health and cognitive function. So, if we can stop the androgen production, then we can simply concentrate on glucocorticoid replacement. This can be usually in the form of hydrocortisone. And we would anticipate that the dosage we would use clinically would be lower than that required to suppress ACTH. So there are various ways of reducing androgen production. I refer back to this schematic diagram of the hypothalamic pituitary adrenal axis. So currently we head to what is required for simple replacement. They are modified and delayed release. The alternative to this is to develop drugs that are able to either stop the production of ACTH or stop the action of ACTH. So, for example, you can develop what's called CRH receptor 1 antagonists. So this is the receptor that CRH acts on in the pituitary gland to stimulate ACTH. So an antagonist to block this receptor will essentially reduce or block the production of ACTH. Another way is to be able to mop up or clear circulating ACTH in the form of monoclonal antibodies. And the final sort of way is to stop the action of ACTH, to develop ACTH receptor antagonists that focus on blocking the receptor to ACTH. So apart from this, there are other methods to reduce androgen production, such as adrenolithic agents or drugs that would inhibit adrenal enzymes. So this next slide, I've broadly shown the drug discovery pathway, which broadly is separated into the discovery phase, the preclinical phase, the clinical phases, and ultimately approval from governmental regulators for use in patients. So I've listed a number of drug classes and where they are in terms of the stage of drug discovery. I'm now going to focus only on the three at the top because these are the ones that reduce ACTH levels or the effect with the aim of reducing androgen production. Just a sort of reminder, if you block ACTH completely, this would reduce androgen production, but you will still require continuation of glucocorticoid replacement. So briefly, studies on the CRH receptor 1 antagonists are underway. They are in phase 2 and phase 3. Now, there's no formal results, but these results have been published in abstract form, as well as at various conferences. And the results look very promising in that treatment with CRH receptor 1 antagonists are able to significantly reduce ACTH, 17-OHP, and androstenedione levels. So I want now to turn your attention onto the ACTH receptor antagonists on some work that we've been doing over the last 10 years on this receptor, and work that was funded in part by IFCA. So this is a schematic diagram of the ACTH receptor, which is also known as the melanocortin-2 receptor. This receptor only interacts with ACTH and is solely responsible for the action of ACTH in the adrenal gland. Ordinarily, the ACTH will bind this receptor along with its accessory protein MRAP, stimulating the production of cyclic MP, which is the way this receptor activates. And in adrenal cells, this leads to steroid production. So blocking this receptor will completely block the effects of excessive ACTH. This is why various groups around the world have been trying to develop peptide antagonists to block this receptor. And another way of blocking, as I mentioned, is regarding monoclonal antibody therapy to try and mop up circulating ACTH. One of the major drawbacks of the peptide antagonists and the monoclonal antibody therapy is the need for parenteral administration. With IFCA funding and also funding from the Medical Research Council in the UK, we've been characterising and developing ACTH receptor small molecule inhibitors with the aim that these could potentially be administered orally. So we screened 200,000 compounds of small molecules to assess whether they were able to block the action of ACTH in cells that express the receptor and its accessory protein. So we measured cyclic AMP as a readout. And out of these 200,000 small molecule compounds, we identified 208 that was able to suppress MC2R or ACTH receptor activity by over a half, whilst having no effect on another G protein coupled receptor, the beta-2 adrenergic receptor. So we worked on these compounds to look at the ones that look the most promising. And I'm just going to show you one of these compounds that we've called compound 4. So the top panel shows a dose response curve. So at varying doses of ACTH, you see different degrees of cyclic AMP generation. And in the presence of compound 4, you get this rightward shift of the dose response curve. And you see no effect on the other melanocortin receptors. So these are family members in the same family as the ACTH receptor or the melanocortin 2 receptor. And this is regardless whether you stimulate with alpha MSH or ACTH, demonstrating that compound 4 is a very selective compound that does not seem to interact with its family members. So using funding from IFCA, we have developed a mouse model to mimic the effects of excessive ACTH in humans. So in these mice, they receive a pump that delivers a high amount of ACTH for over a 2-week course, and then we will carefully phenotype and assess these animals. So this is just some preliminary data to show you that this model does cause adrenal enlargement, adrenal hyperplasia in a dose-responsive manner, as well as hypertrophy, which is exactly what you would see in congenital adrenal hyperplasia. So we aim to use this animal model to test the compounds, including compound 4 and its associated alternative structures, to see whether these compounds would be able to suppress and change this hypertrophy phenotype. So in conclusion, I would say that we have successfully identified a highly specific ACTH receptor antagonist in vitro. The properties of compound 4 is suggestive of potential oral bioavailability. There's still quite a lot of chemistry that we need to do regarding that compound. This would be highly advantageous in the long term. This would allow block and replace therapy, which would be a sort of new concept of treating congenital adrenal hyperplasia, as well as potential short-term suppression of adrenal hyperplasia. As the sort of previous speaker mentioned, it could potentially also be a treatment for something like TART. So generating an ACTH excess mouse model to test compounds it has been really useful for us so that we can test these compounds that we have discovered. Overall, there's quite a lot of interest in the pharmaceutical world to develop compounds to suppress ACTH. And I, for one, think that in something like 5 to 10 years' time, we'll be seeing some of these compounds come to market. So I would like to acknowledge my team, past and present, as well as our collaborators. Most of the discovery work was performed by Mashal, my previous PhD student. The mouse work was performed by Nini, and Dylan's a new PhD student in my lab, and Nini and Dylan will be the ones taking over this work. And I'd like to thank IFCA for their generous funding, and I'd like to thank everyone for their attention. Thank you.
Okay, thank you very much, Lee, for this wonderful presentation. I can comment. I can ask you some question.
Sure.
Can you comment on adrenal crisis in this patient? Because, you know, the group of AD Klassen have demonstrated that the accumulation of some steroid before the 21-hydroxylase can have some glucocorticoid activity and then prevent from adrenal crisis. So what do you think?
So I think that there will be a certain group of patients that would truly benefit from ACTH inhibitors. I mean, there's always a sort of discussion whether those individuals that, how do you put it, do have some cortisol secretion, so there's a partial block, would you treat them? So I think that the information is basically requirement of understanding long-term outcomes of those groups in some ways, because if you find that actually treatment of those groups do have better sort of metabolic profiles long-term, then actually it is worthwhile treating. I mean, I think that you are fully aware that when you treat them on ACTH inhibitors, you're going to monitor them quite carefully in terms of what their outputs are, as well as you do need to sort of continue glucocorticoid replacement therapy.
Okay, thank you very much. There is some question in the chat. When do you think a long-release treatment will be available? I think that's for Richard Ross. When is this treatment likely to be available?
Well, I mean, sort of like there's so I think there are three sort of possible routes to treatment in terms of ACTH, what I call ACTH inhibitors. But actually, we're talking about a combination of CRH receptor 1 antagonists, which actually already is in phase 2 and 3 clinical studies. So I can imagine that won't be, that long before it's assessed and the data is out. But, having said that, I'm not part of that study. And, I think Richard Orcus will have a much better view about the timeframes of that. But I think the data that they've presented look very good. I think when it comes to ACTH receptor antagonists, we still have quite a way to go in terms of its developmental phase and then preclinical, clinical trials type situation.
Okay, just 3 questions. What is the potential benefit of ACTH monoclonal antibodies over ACTH receptor blockers? That's from Martin Koos.
Yeah, it's difficult to know, isn't it? I think I prefer the idea of an ACTH receptor blocker, just because I work on a condition called familial glucocorticoid deficiency, whereby some of these patients have a natural mutation in the melanocortin-2 receptor. And I, for one, know that those individuals have isolated glucocorticoid deficiency. So, we have some human data to say long-term, if you block the melanocortin-2 receptor, your outcomes are not terrible. Now, I think that the antibody aspect is slightly more unknown in that respect, and it needs to be very specific, and the only data is preclinical, so it's difficult to say about that.
Okay. A question from Leonardo Guasti. Is your compound able to cross the membrane? Meaning, could it be used for familial glucocorticoid deficiency patients via rescuing of the mutated receptor?
So Leo is a colleague of mine that works in the department, and we often have these discussions. I don't know the answer to that. Some of the compounds that we've discovered through this process are very lipophilic, so it's a sort of aspect that we're sort of currently looking into.
Okay, and sorry. Okay, 2 questions from Eddie Klassen. Do you have information about teratogenic side effects? She means like pregnancy rate in animal experiments under treatment with ACTH antagonist. And do you have some data on the development of adrenal rest tumors in these mice?
Yeah, so it's really interesting that sort of like my talk came straight after Heidi's talk. So the testes is one of the organs that we will look at in these mouse models. But bear in mind, we've only stimulated, we've only delivered 14 days worth of ACTH in these mouse models. So I don't know how it's a long enough time for the sort of development of TARTs. So the other question, what was the other question?
On pregnancy, teratogenic effects, so the same. Yeah.
We don't know that answer at all. It's not something that we've done yet. So, but it's a good question. As you know, with all treatments, we need to know what the side effects are.
Last question, Michelle. Yes, it's okay. The timekeeper. Okay.
Yes. Last question. Short question, short answer.
Okay.
Thanks.
A question from Tule Guram. In mice with adrenal hyperplasia induced by ACTH pump, is that hyperplasia zone specific, and are the androgens elevated in those mice?
Yeah. So the majority of the hyperplasia is fasciculata. That's the major zone that is expanded within this mouse model. So androgens are slightly difficult when it comes to mouse models because they lack CYP17. So they don't secrete androgens in the same way as humans. So it's a sort of slightly difficult sort of, it's a slightly more difficult thing to measure, if you see what I mean. But so we're doing it sort of histologically more than anything else.
Okay. Thank you very much.
Thank you so much.
Thank you, Lee. Wonderful talk and wonderful open windows to new treatment.
Yeah, I think it's an exciting area, but I'm slightly biased. Thank you. Bye.
Yes, but we like your bias. And Ivka liked it as well. So thank you, Anne, for moderating the session and the Q&A session.